Hello everybody, welcome back to another episode of STEM Nerds. I'm Jason. I am so glad you're here today because in today's video we have to talk about how gene expression is controlled. In previous videos we've talked about going from DNA to RNA and transcription and from RNA to proteins and translation. I'll put those videos in the upper right hand corner as well as in the description below. But today we really have to focus on how we control these processes because they can't just run all the time. So let's take a closer look. I got the whiteboard out. Hopefully that'll help us understand it a little bit easier. Thanks for being here. All right, so here we go. What we have is a gene. We have a gene and we have a control region here. And the first thing we're going to talk about are genes that are inducible. In other words, they're normally off, but they can be induced. They can be turned on. We're going to talk about how that happens. And so we've got some things that we have to identify. We have a gene of interest here. Then we have the control region that consists of an operator, and an operator is where a repressor can bind. And then we have the promoter, something we've already discussed, and that's where RNA polymerase can bind. Well, in an inducible system, the gene is normally off, but it can be turned on. And in this particular case, we're going to be utilizing a repressor to control gene expression, and that means that this gene is under negative control. Whenever you see a repressor being used to control gene expression, we're talking about negative control. Okay, so normally the repressor is bound to the operator, and that repressor is going to be in the way. It's going to block the ability of the RNA polymerase to access the gene, and so right now gene expression is turned off. But we can induce gene expression, and the way that we do that is we have the repressor here, but we see the repressor is not bound, and the reason it's not bound is because we have an inducer. And so this inducer binds to the repressor, prevents the repressor from being able to bind to the operator, and now, because of that, RNA polymerase doesn't have anything standing in the way. It can get to the gene, and it can begin transcription. So this is an example, again, inducible system, normally off, but can be turned on. And the way that it's turned on is when an inducer shows up, binds to the repressor, releasing it from the operator, gene expression is on. Now here I want to show you another example. Again, we're using a repressor, so we're still under negative control, except this is a repressible system, which means the gene is normally on. So this is a little bit different. Normally the repressor is not bound to the operator and the RNA polymerase has full access to the gene. However, in a repressible system, there's a feedback mechanism that allows for the production of a co-repressor. And that co-repressor is like a little piece of glue, and that little piece of glue glues the repressor to the operator, blocking gene expression, and it's a way to shut it off. And so in a repressible system, under negative control, usually on, but it can be turned off. You know, before I get into the next potential scenario, I do want to mention that there are some genes that are constitutive genes, and that means these are genes that are pretty much always on. And so they're generally not being turned on or turned off, whether it's by a repressor or an activator, which we're going to talk about next. All right, so there is another component to this, and that is positive control. And so gone are the repressors. There's no repressor anywhere on this board. What we have instead is an activator. And when an activator is used to control gene expression, that is positive control. And usually in positive control, that's going to be an inducible system. In other words, normally the gene is off, like we saw before, and it can be turned on, but for a different reason. So in an inducible system, an activator, if the gene is off, is not going to be bound, and therefore RNA polymerase is also not going to be bound, and so we don't get any gene expression. I want you to think of the activator as like a motivational speaker, encouraging RNA polymerase to bind to a promoter. The only way that's going to happen is down here we see a situation where this gene gets turned on, and that is the activator is going to need an inducer. The inducer here binds to the activator, allowing it to bind to an activator binding site. Once that happens, that's the motivation that the RNA polymerase needs to bind to a promoter and begin the process of transcription. And so here we have a second type of inducible system. In the previous example, it was inducible because we were removing a repressor with an inducer. Here, also inducible, but under positive control, and the inducer is allowing the activator to bind to the activator binding site, allowing for gene expression. Okay, so we've talked about positive control, negative control, how to use repressors, activators. Well, let's put this all in a practical example, and that is something called the lac operon. This is a series of genes that are used by E. coli and other microbes to break down lactose when it's present in the environment. So an operon consists of the genes. In the lac operon, there are multiple genes. For simplicity here, I just put genes here. So you've got the coding region, and then you have the control region, which consists of our activator binding site, something we've already learned about, promoter, and operator. And so one of the things that we have to know about the lac operon is for microbes that possess the lac operon, they generally prefer glucose over lactose. And as long as glucose is there, there's no reason to turn this thing on because it's going to consume its favorite food source and not resort to something that it doesn't like as much. So we have this gene structure here, this operon structure, and now we've got these 4 different examples. And I want to kind of run through and look at how both positive and negative control plays a role in gene expression. And yes, the lac operon is under both positive and negative control because both activators and repressors are being utilized to turn this operon on or off. Okay, that being said, what we have to do is wrap our heads around exactly how this operon is being controlled. So I'm going to give you an example here. Example number 1, we have a situation where in the environment glucose is present, but there is no lactose. First thing you should be asking yourself is, okay, is this cell happy or sad? Well, if glucose, its favorite energy source, is present and lactose isn't, that sounds like a pretty happy cell to me. And indeed it is. And then the second thing is, okay, well, if it has glucose, why would you ever turn on a bunch of genes that are made to break down lactose when there's no need to? Not only that, lactose isn't even there. So this operon, these genes are not going to be transcribed, and the operon is going to be off, and there's a couple reasons for that. One, the activator is not bound in this situation. We'll get into why in a second, but right now, with glucose present and no lactose, the activator is not bound, which means we talked about the activator is kind of like that motivational speaker that allows RNA polymerase to bind, and that's not happening because the activator didn't bind. And right now, the repressor is blocking the way to the genes anyway. Okay, so that's situation number 1. Situation number 2 is we have glucose present, that's awesome, but there's also lactose. Okay, so now the cell has a choice, but we already know the cell is going to choose the glucose every time. And again, if glucose is present, why would we turn this operon on? Well, we wouldn't. But I want you to pay attention to what the differences are between scenario number 2, the one that we're going over now, and the one that we just did. And so environmentally, lactose showed up. That is the only change between these 2 scenarios. Okay, what changed in terms of our control region? Well, the activator is still unbound. Because of that, the RNA polymerase still unbound to the promoter. What changed once lactose showed up is the repressor is released from the operator. That means, and something you should know, is that the presence or absence of lactose is going to control whether or not the repressor binds to the operator. When lactose was missing up here in this first example, the repressor was bound. Now that we've taken the lactose away, now that we've added the lactose, the repressor has been released. Okay, now we go to our third example. We got a problem here. We've got no glucose and no lactose. So do we have a happy or sad cell? Well, we got a pretty sad cell because not only is it the favorite food source isn't there, it doesn't even have a backup. And again, practically speaking, why would we turn this operon on and transcribe genes for a sugar that isn't even there? But again, let's pay attention to what has changed in the environment relative to what has changed in our control region. This is the first time where glucose is missing. In the previous 2 examples, glucose, activator is unbound, glucose, activator is unbound. But now, no glucose. And look what's happened. The activator binds to the activator binding site, which means, if you're keeping score at home, glucose controls whether or not the activator is going to bind to the activator binding site. Now in this particular case, it doesn't matter. The activator has bound, which allowed RNA polymerase to bind, but because we have no lactose, just like we did in the first example, our repressor is bound, it's in the way, it's not going to allow RNA polymerase access to the genes on the other side. Last example, down here, we have no glucose and we have lactose present. So our cell is not exactly happy, but at least it's not in the same situation that it was in previously. There is a food source present. And this is the only scenario in which the lac operon is actually turned on. Why? The absence of glucose, just like in the third example, means the activator is bound. That in turn means that RNA polymerase can bind. And now, because we have lactose, just like we did up here in the second example, the repressor is released. And so that roadblock to accessing the genes on the other side is now gone. And so in this last example, that's the only time that the lac operon is going to be turned on. So we've learned about positive and negative control and how we can control gene expression utilizing repressors and activators, but there is another way. However, the way that we're going to be talking about here is not controlling whether or not we're turning genes on or off. It's after transcription has already occurred. So this is going to be post-transcriptional. Now, in a previous video, we talked about exons and introns. If you remember, the exons are expressed and we make proteins from those. The introns are not expressed. They are the parts of the messenger RNA that have to be removed before the messenger RNA can leave the nucleus and go out into the cytoplasm and be translated into protein. At the time, I told you that when I was in school, we were told that the introns were just junk DNA and that they really didn't have a role in any way, shape, or form. Well, some smart people went out there and questioned that, and they found out introns actually do have a really important role overall in the cell. And what they do is they're involved in control as well. And so let's say you have, this is up here, this is our primary transcript, our messenger RNA that was transcribed from DNA. We remove all the introns, we splice the exons together. Exons leave the nucleus, they get their cap and their tail, and now this messenger RNA can be translated into protein. Well, what happens if you have enough protein? How do we stop this message? This recipe is out there. And ribosomes don't necessarily know, oh, hey, we've got enough, I can go ahead and stop. They just keep reading the recipe and making more of the protein unless something tells it to stop. And that something sometimes is those little intronic regions. And so there are other types of RNA out there, and just as a very simple summary, we've talked about messenger RNA, we've talked about transfer RNA, we've talked about ribosomal RNA. But you will see from time to time RNA with other letters. And so some examples, RNAi for RNA interference, siRNA, small interfering RNA, microRNAs. When you see letters before or after RNA that aren't the big three, generally speaking, they're going to be involved in control in some way, shape, or form. And so this example that I'm showing you here is a type of RNAi, RNA interference. And what happens is, remember that the messenger RNA is made up of A's, U's, C's, and G's, and it's single-stranded. Well, parts of an intron can leave the nucleus. In fact, when I was in school, we were told that exons exit the nucleus and introns stay in the nucleus. Well, we can't say that anymore because we know that introns, small parts of them, can leave the nucleus, go out into the cytoplasm. Using base pairing rules, A's, U's, C's, and G's, they can bind to the messenger RNA that's out there. And when this happens, it forms little parts of double-stranded RNA. Cells, generally speaking, don't like that. It either means they're infected by a virus, or we have this situation, which is a feedback mechanism telling the cell, hey, you know what? We've got enough of the protein. We don't need this recipe anymore. It's time to shred it. It's time to stop it. And then what will happen is once these double-stranded RNAs are out in the cytoplasm, there will be enzymes that come in, like Pac-Man, and they gobble up the messenger RNA, shredding the recipe so that the protein production stops because the protein production is no longer needed. So these introns are super important, and for the longest time, scientists have been looking at mutations in exons, the expressed regions of the DNA that lead to messenger RNA, to see if there is something wrong in terms of the genetics for a particular organism. Well, now we're starting to look a lot more at these introns because think about it. If these introns are mutated and those intronic regions that are supposed to control really translation downstream cannot bind to their messenger RNA target, you could wind up getting more proteins than you actually need, and that can lead to all kinds of metabolic disease. And so these introns, they make up the majority of our DNA. Scientists have to go back through and figure out, okay, if there's mutations in the introns, what exactly is happening and what is the impact metabolically on those cells. All right, everyone, so that is a brief summary of how gene expression is controlled utilizing inducers and repressors and even little pieces of RNA. As always, thank you so much for being here. We'll see you next week.